Freezing human iPS cells. This protocol video outlines the cryopreservation of one well of human iPS cells cultured in a 6-well plate in 2 cryovials. To obtain the best recovery upon thawing, freeze healthy, actively dividing, undifferentiated cells. Factors such as passage number, proliferation rate, and differentiation in the culture that is frozen can have an effect on the successful thawing of human iPS cells. Step 1. Enzyme Incubation. Aspirate the medium from one well of human iPS cells to be passaged. Next, add 1 milliliter of collagenase 4 at a concentration of 1 milligram per milliliter to the well. Incubate the plate for 5 minutes at 37 degrees Celsius and 5% CO2. Step 2. Removing the enzyme. After the 5-minute incubation with collagenase, observe the cells under a microscope and note the edges of the colonies should appear more defined and slightly lifted from the edge of the plate. If the enzyme does not appear to have had an effect on the colony edges, the cells can be incubated again for another 2 to 3 minutes. Aspirate the collagenase enzyme from the well of cells and add 2 milliliters of human iPS cell culture medium to the well. Step 3. Cell collection. To collect the cells, use a 5 milliliter glass pipette. Glide the end of the pipette across the surface of the plate while slowly expelling the medium to gently detach the cells from the plate. This first harvesting step can be repeated 2 to 3 times, but be careful not to over-pipette the detached colony pieces. Transfer the cells to a 15 milliliter conical tube. Human iPS cells proliferate in an undifferentiated state when passaged in small colony aggregates, but not as single cells. It is important to mechanically break the colonies into appropriately sized colony pieces during the passaging process to ensure proper attachment and proliferation. Using a new 5 milliliter glass pipette, add an additional 2 milliliters of HIPS cell culture medium to the well and repeat the scraping technique to harvest the remainder of the cells from the surface of the plate. Transfer the cell solution to the same 15 milliliter conical tube. Centrifuge the cells for 5 minutes at 200 times G. Step 4. Prepare cryovials. While cells are being centrifuged, label 2 1.5 milliliter cryovials with the cell line, passage number, date, and your initials to ensure the cells are frozen as quickly as possible. Any other information about the culture or experiment should be recorded in a lab notebook. Remember, this information will likely be erased by ethanol upon thawing. If you are using printed labels, confirm that the labels will not fall off if submerged in liquid nitrogen. Be sure to completely loosen the caps on both vials. Step 5. Freeze cells. To begin, remove the supernatant from the cell pellet. Gently resuspend the cells in 2 milliliters of 1X CryoStem cell freezing medium. Remember to work quickly once the CryoStem cell freezing medium is added to the cells. Pipette the cell suspension to mix evenly. Add 1 milliliter of cells to each cryovial. Cap cryovials tightly and place in a room temperature isopropanol freezing container. Place the isopropanol freezing container in a -80 degrees Celsius freezer overnight. The isopropanol freezing container will freeze the cells at a controlled rate of 1 degree per minute. The following day, transfer the cryovials to a liquid nitrogen tank for long-term storage. Vials can be stored in either the liquid or vapor phase of liquid nitrogen. If you are storing for long-term storage of important cells, store the vials in a rack that is seldom accessed or at the bottom of the rack to avoid repeated and unnecessary temperature changes. Additionally, when thawing a vial of cells, it is good practice to expand and freeze additional vials to maintain a sufficient supply of low-passage cells for future experiments. It is also a good exercise to routinely freeze extra human iPS cells to add to the frozen stock. For a library of Stemgent video protocols or to download a PDF of this protocol, please visit stemgent.com.